Our next speaker is Pascale Vonaesch from University of Lausanne in Switzerland, and she will present pilot study of the Microbiota Vault. So please say hello to Pascale and go on.
So hello, everyone. Thanks a lot for having me and for giving me also the opportunity to present in the name of the whole launch team, the Microbiota Vault launch. I would like to start with showing you, if it works. Yes, here. I would like to start by showing you this picture where you see that in the last 50 years, there was really a decrease in infectious diseases. And at the same time, on the right side, you see that during this time, there was also an increase in non-communicable diseases. And so this dichotomy is believed to have to do with the use of antibiotics, which were introduced shortly before. And it seems that basically while this helped to curb many of the infectious diseases, these changes in the microbiome also predisposed basically many people to develop these non-communicable diseases. So this has really nicely been discussed in a book by Martin Blaser that I think spoke yesterday, and that wrote a book that is called The Missing Microbes that I really invite you to read. I would also like to mention that at the same time, there has been quite a lot of research where other researchers, including Gloria Dominguez-Bello, that is also here today, have looked at the diversity of the microbiome in children and adults from countries where there's less basically like industrialization. They're often called like non-westernized or more traditional communities, and also children from the US. And what they observed is that they could see that there was a steady decrease in diversity of the microbiome, which is another hallmark that is often associated with these non-communicable diseases. So this has then been looked at in more details a bit later by another group. They took many different countries and could really see that there seems to be a general trend that by increasing westernization, there is a decrease in microbiota diversity. So this really raised a lot of interest and was also like of big concern. And subsequently, people also started to look at the different bacteria that were changed, and they could see that, as an example, in the industrialized communities, as they defined them, there was an increase in different taxa, including the Bacteroideaceae and also the Verrucomicrobia, while in more so-called traditional communities, there was an increase in other bacteria, especially in the Prevotellacea, the spirochetes, and the Succinivibranacea. So it really seemed pretty easy, black and white. Either you're having a traditional lifestyle, you have certain bacteria, you're industrialized, you have some others, and this predisposes you to non-communicable diseases. But as you can already see in the title, it's often not as easy as that, and it's already quite difficult to really define what do you mean by traditional and what do you mean by industrialized. So we have recently published with my own group a study in Ethiopia. So this is a pastoralist community living in the Horn of Africa in the Ogaden region. And they have a very specific diet where they drink a lot of camel milk, have also quite a lot of wheat that they eat, but very fiber poor diet. And what we observed was quite interesting because even though this population should probably be considered traditional, there was both actually low levels of Bacteroideaceae, which are specific for the industrialized nations, but also low levels in Succinivibranacea, which are basically more characteristic for traditional communities, and the same also with Prevotellacea that I didn't depict here. So this really showed that it's not as easy as that, it's not black and white, but there really seems to be bigger changes in the human microbiome that are really linked to many dietary practices, amongst other factors. So to really showcase also that the microbiome differs across the world, I would just like to show this publication that recently came out by another group. And what they did is that they looked at the so-called healthy microbiome by looking at centenarians. And what they did is that they looked at centenarians across the globe and could show, as you see here with the different colors, that the healthy microbiome of these people really differed in the different geographic regions. And this per se is not really something that would be unexpected or worrisome, but I think when it's basically becoming a bit worrisome is when you look at this other map that comes also from a recent publication where they looked at the availability of genomes from bacteria that have been sequenced across the world in the intestinal microbiota of humans. And as you see, there's big gaps actually, especially in low and middle income countries, which basically means that we know that the healthy microbiome has really changed across the world, but at the same time also that we only know about certain hotspots in the world, how such a microbiome normally really looks like. So all these reasons combined has then led Maria Gloria Dominguez-Peya and Martin Blaise a few years back to initiate the Microbiota Vault. I believe Gloria also already said a few words about it yesterday. But the reasoning was really that the human-associated microbiota play a significant role in the biology of humans and in human health, that basically global developments of westernization and urbanization can lead to irreversible impact on the global diversity of this human-associated microbiome. But at the same time, we only know a fraction of the global microbial diversity today. So we basically risk to really lose basically this diversity before we even understand if it's important for our health. And therefore, it really is obvious that there's a need for a global collection of the microbiota and for the safe storage and preservation. So the goal of the Microbiota Vault is really to have a Noah's Ark for human benefit. So basically to work with regional working collections that submit diverse microbiota samples, store them in a safe, politically neutral and stable location on behalf. And this on behalf is really important because the Microbiota Vault doesn't own any of the samples. They're basically just keeping them safe. And the Microbiota Vault would then also enable research, not to itself, but really enable it, so that ideally we could then in the future also restore basically like disturbed microbiomes and basically like contribute hopefully also a little bit to human health. So the Microbiota Vault had a feasibility study that was really promising. And after that, there was a launch that was planned. And that's where Adrian Egli, Nicolas Bokulic, and myself also joined the team and started to work on this launch. And we all three have different expertises on the scientific side and are supported also by Gloria Dominguez-Peya, Dominic Steiger, and Manuel Fankhauser, which really work hand in hand with us and also bring in a lot of expertise in the overall management, in fundraising, and in all the legal and regulatory aspects. So the launch is structured in 7 different work packages. The first, which is the actual Vault initiative, which aims to look at the governance structure and associated legal framework to store the samples on behalf of the collections. The second, that is to validate the SOPs. So as an example, to have standard operating procedures for the collection storage of fecal samples. Work package 3 and 4 aim really at playing through the local collections and the sample pathway. We'll get back to that in a minute. And then work package 5, 6, and 7 are really aimed at actual storage, at data and annotation, and also at sequencing of these samples. And we'll get back to that in a little bit as well. So the general structure of the Vault is really to have these collections associated with a given set of metadata and to store all of this together in an openly accessible space. If the collections agree, there would also be sequencing that is basically proposed, which is then also openly accessible and really allows also to see what is present basically in the samples that are stored. And this sequencing would then be analyzed for all the samples in a standardized way using QIIME2 that was co-developed by Nicolas Bokulic and would thus allow to really see what is available in this Vault. So for the sample pathways, I think if someone thinks about it, on first sight, it seems easy to say, well, we just go and collect the samples and bring them back in a safe place. But there's many different aspects that have to be controlled for it, starting already with on-site sample collections to be sure that basically the samples are in an optimal state. Also everything related with the logistics, the sample shipment, until finally they get in the lab where we have to control for their quality, store them in an optimal way, and finally also set up this database where they can be integrated. And for now, we have for the launch samples from 4 continents. So it's 6 collections that are included in the launch, with a collection from Switzerland, from Laos, from Ethiopia, from Ghana, from Puerto Rico, and also from Brazil. And right now, half of the samples are already in Switzerland and have already been started to be incorporated in the Microbiota Vault. So another aspect that is quite important for the launch is also this establishment of the best preservation methods. And what we have focused on so far is really to have methods that would hopefully allow to then also regrow a maximum of bacteria from these complex samples. So what we did here is that we tested a set of different cryoprotectants, reducing agents, and storage temperature, with the questions to have something that really allows for a maximum of recovery of different bacterial strains. This is also quite cost effective so that they could be used in different settings. And that is also easy to use in the field, as we want this method really to be accessible to everyone throughout the world. And the idea is to have then this standardized SOPs published so that everyone can have access, can sample basically like in places that they think are of interest, and then basically contribute these collections to the vault. And to set up these different techniques, we have tested so far 15 stool donors, 5 from babies, 5 from children, and 5 from adults, as they have different microbiotas. And we assessed, as I said, then the best storage conditions. And we also wanted to assess at the same time which methods would be good to basically test incoming samples for their quality, so that we're sure that everything that goes in the vault really has the quality that is needed to basically deserve a long-term storage. So we did so in a systematic way using different agar plates and also different freezing methods. We took the original sample and then sequenced it to see what's really in there. At the same time, we also spread the sample on different agar plates, and this was really to see how much of the sample is still alive and how much of this bacteria we could then basically like regrow on plates. And at the same time, we then also froze the sample in different conditions, as you can see here, using glycerol, DMSO, and palladium beads as a reducing agent and different combinations thereof. And we then also stored basically the samples either at liquid nitrogen or in a -80 freezer. And 1 year later, we then reactivated the samples again, spread them again on the same plates, recovered what was still alive, and basically sent it for sequencing. And right now, we just finished the follow-up of all the samples. All the DNA is extracted. We've made about 2,000 samples, and the sequencing is, as we speak, ongoing. So we hope that in a few months, we can also then share with all of you which of the techniques are really the best to preserve a maximum of the different strains. And of note, we also tested lyophilization as an alternative method, which would then obviously need some more infrastructure, but could be easier to store in different contexts. So at the same time, we also work on minimal metadata and the sequencing. This is work that is led by Adrian Egli, Nicolas Pokulic, and their teams. And I think we will hear more about the metadata also from Anton Lavrinenko later today. And at the same time, we're also working on this structure to basically sequence, store, and basically take together all the data to really start the nucleus of the actual vault. So in conclusion, I hope I could show you that we really need a Noah's Ark for the human beneficial microbes, and that it's also important to basically have methods that will allow us to later reactivate bacteria from complex samples, including also the dark matter, meaning like bacteria that we maybe don't even know today yet. I also hope that I could show you that standardization of collection, metadata, preservation, quality control, and biobanking is really crucial, both for having good quality samples, but also for reproducibility across different methods and also research. And finally, with the Microbiota Vault and also in the launch, we also want to foster microbiome research across the world and really have this launch as a first step towards a global effort. So I hope that this gave you a first glimpse of what's going on in the launch. If you're interested, I also invite you to watch this small video that has been produced by Razor and shows a bit more in detail what we actually are doing right now. And with this, I would just like to thank everyone that was involved, especially Marty and Gloria, but also Eugen Theo from my lab that was leading the different aspects of the wet lab work that I've shown. Adrian Egli and Nicolas Pokulic, which are on the scientific team with me, Dominik Steiger, Manuel Fankhauser, all the local working collections that contribute, the Microbiota Vault Consortium, and also especially our funders that really believed in this idea and supported us for the launch. And thanks you a lot for your attention.
Thank you very much, Pascale, for your presentation. It's very nice.